Welcome to Cycle 2, Inheritance of Sameness. I will be with you from Cycles 2 through 5, and then you're back with Beth. Before we start lecture, we have a biology mentor representative that would like to come up here and make an announcement.
So hello guys, so as you guys know, we finished Cycle 1, moving on to Cycle 2, and the Biology Mentorship Program is going to hold a workshop today at 5:30 at NCD 340. So just go up to the third floor, go to the end of the hallway, and you'll find us there at 5:30. So come through, we'll be going over the outcomes and a lot more. It's going to be a lot of fun, and it's going to be a great review for you guys. So come through, I hope you see a lot of you there. Thank you.
Thank you. This is a great workshop, great mentorship group. I would absolutely take advantage of them and really attend these workshops. Most students that do find it very beneficial, very helpful, so please do that. And they're just an amazing team that are really out there for your success. Okay, thank you. Before we start lecture officially, I have some quick announcements. Number one, please check announcements, messages on OWL, and your UWO email regularly so that you don't miss important announcements. This is the only way for us to communicate with you. We have no other way to communicate with over 1,500 students. We want to be assured that if we post an assignment, if we post an announcement or send you a message, that you are receiving it and you're checking it regularly. So if there's something important that we have to communicate with you the day before the term test or the day before the final or the day of the quiz, it's just nice to know that you're aware that messages will be coming through and they could be very important ones. This is really pivotal to your success too. Don't miss messages that are important. For example, I posted an announcement on September 13th regarding quizzes, assignments, test document. There was a document attached to it as well. I hope that all of you received it and all of you have looked at it. So these are important things that come up, and please don't miss them. OWL quiz for Cycle 2 is due September 23rd at 10:30 a.m. and September 24th at 10:30 a.m. depending on your section number. Grade and feedback will only be provided late Tuesday. This is to ensure fairness in the course. So, for example, the first group, if they did the quiz and they got 100%, then they know all the correct answers. So they can pass that on, not that they would, but we need to maintain that kind of fairness. So marks are not released until everyone has completed the quiz. Some OWL quizzes, not all, and within an OWL quiz, some questions are set to a randomization setting, meaning that question 1, for example, if it's on DNA replication, everyone will receive a question on DNA replication, but it'll be different. So if you look at your peer and they are writing a quiz that looks different than yours, it's because that's the question they received in the randomization. This is to allow for variance in the quizzes and to get more questions out there. They're all exactly equal level of difficulty. Future OWL quizzes. Historically, this course has always had OWL quizzes due 24 hours before game day. We are going to make an exception this year and make it available to you 48 hours before. There was actually just one student that approached me and said that Sunday was very inconvenient. Sunday is a day where you go away with family or friends and you may not have internet access. I thought there was a legitimate concern there, and so we went ahead and made that change. And sometimes it just takes one student, and we respect that student's opinion, and we do implement it in the course if we feel that it's something we can implement. Sometimes it's very difficult, but this is something that we could do. Skill development assignment is due before your first lab session. So many of you have already completed it because you've had your lab already. Please make sure that you go to the skill development tab, go to module 1, and all the directions are there. It takes about 90 minutes to do the assignment, so please make sure that you keep enough time. Your first 3 MindTap quizzes are due September 24th at 11 p.m. Set your MindTap to Eastern Standard Time so that if you are receiving alerts, you receive them at the proper time. Please ignore all MindTap grades. Don't worry about what MindTap is telling you what your grade is. As long as you complete 80% of the quiz, you will receive a mark of 100%. So if your MindTap quiz is telling you that you got 0% on that quiz, it's okay. In our books, that translates to 100% because you participated. It's participation only. There's no reason why everyone in this class should not be getting that 3%. It's almost a free 3%. Make an effort, do the quiz. It really is A+ for effort. The overview page on OWL, that will also contain some important announcements, due dates, so do check that as well. So there are many avenues. I'll post them as announcement. Messages on OWL. Messages, you have a message inbox on OWL. You need to check that. I'm not always going to send you announcements because announcements go to everyone that's attached to this course, all administrators. I don't think administrators repeatedly want to receive all kinds of clutter in their email from this course. If I go through messages via OWL and just hit students, then it'll only go to you and we don't clutter everybody else's inbox. So please check OWL. I will always attach it to your UW email, but it doesn't hurt. As soon as you open OWL, go to your main page. It says how many new messages you have. Just click on that. Make it a daily habit to check your message on OWL as well. I just want to go over a little bit about MindTap really quick to make your life easy. How many of you watched the YouTube video that was posted? If you haven't watched it, please look at that announcement from September 13th. The link is on there, and you can take a look at that video. It's 9 minutes. It's very informative on how to navigate an OWL, how to do your quizzes, how to submit them, where to look for correct answers, and where to click so that it takes you directly to the page and the section in the chapter where the answer is explained. So that's really helpful. Instead of you're like, the feedback doesn't really help. I need more explanation. I need to understand this better. How do I go? Where do I go? How do I rummage the textbook? Do I search? How do I do this? No, you just click on the incorrect answer and it'll take you to this page that shows you where the correct answer is and how it's described. It's very helpful. It's a good study tool. So when you first open up your MindTap, it looks like this. Except this is the instructor view, so bear with me, much of it is the same. You look at all these activities. For example, Cycle 2 has 20 activities, Cycle 4 has 7, Cycle 5 has 8. That's a lot of activities. What are these activities? They're essentially assignments type things. So they have practice, questions, test your own knowledge, self-assessment, etc., etc. We lump all of these things and we call them MindTap quizzes. That's what we do, just to make your life easier. MindTap is a separate platform, it's a separate program, it's not OWL, it's not this course. So what we do is, we just said, instead of calling it 100 different names, self-assessment, practice, test your knowledge, it gets too confusing, let's just lump it all and call it MindTap quizzes. So that's what that is. Now having said this is not the number of activities you have to do for marks. So if you actually hit on that and scroll down, you'll see this drop down and it gives you these check your knowledge, self-assessment, there's a timer on it, and students have asking me which one counts for grades, which one am I doing? And the ones that you're doing are the ones that have an orange dot next to them. There's an orange dot next to it, that's the one you're doing. So these ones you can do for practice, but these are not going to count towards your participation marks. Then if you click on Cycle 2, Got It Steady, now all of a sudden you see assessments that have orange dots on them. Those are the ones that count for grades. Those are the ones that you have to do and submit. The due date is written on there and the time that is due is written on there as well. If you really don't want all this extra clutter, my recommendation would be to simply go to Filters and click on Counts Towards Grade. Then, when you go to the drop down, you see the same thing, except all that appears in that drop down are the orange dot quizzes. That's it. So each drop down will just tell you what's due and when it's due and how many activities. So for example, Cycle 1, there's nothing there. There's a lot of readings and practice, but there's nothing with an orange dot, so it says 0 activities. So if you're concerned about what counts for grades, just tell me what counts for grades, then this is the way to do it. I showed you this in welcome lecture as well as a Q&A. I just wanted to mention one thing. You do have MindTap quizzes that are due during the class prep. I know I misspoke when I said that MindTap quizzes are only due between the start of cycle and end of cycle. That's not true. You do have MindTap quizzes before the start of a cycle during the prep. So please just go on there and honestly just participate. Don't stress over it. There's not something I would stress over. You have a lot to stress over. MindTap quizzes should not be a source of stress. They should be a great resource. You don't have them there to make your life difficult. People that do MindTap and do practice, they do find it better in understanding concepts, they get more practice, and they're able to do better on the exam. So keep in mind that that is a great tool. We have in this course what's called specifications grading. This is actually a type of grading that I really think is going to be popular all throughout the country very soon. Very few places are using it right now, and there are actually some big centers in the States that are looking at this and saying, how are you doing this, get us involved, and what's happening. So we're actually leading in this type of assessment. This is actually very beneficial for students. Is it a lot more work for administrators? Yes, it is, but it's worth it because it's worth providing a great student experience, and that's really been a big goal for this course. So what specification grading does is it basically tells you that you don't have to be perfect. And what we mean by that is if you look at this module 1, you have X number of assessments, and then to get level 2 mastery, you can fail 1 skill assessment and 1 advanced skill assessment. You can fail 2 out of these and still receive what's called a perfect score, 100%. And this really does follow our philosophy of what we mentioned in class in the beginning on welcome lecture, you know, the key to success, what success looks like, and you can be perfect, you can have success and reach to the top, but not achieve everything perfectly. And that's the theory that we implement in here too. Not just say, well, success, just fail, but you know, you'll still succeed. Well, I'm failing in this course, but I'm not succeeding. Yes, you have that opportunity. You have that opportunity in skills, you have it in tests, you have it on the finals. So you have, you know, October test is low, we reweigh it. November test is low, we reweigh it. So we give you 3 chances. It's okay. You can actually fail the October. You could do really poorly on the November and still come out with a great mark because we are going to reweigh those marks. It's us saying, you made it there. The success path didn't look like the one that I showed you. It looked like the other one, which is the reality, but you made it at the end. So I think it's important for courses to implement this. If we're teaching that to the students, then we should implement that in the course as well. My office hours, I'm here to help you succeed. My office hours are Monday to Thursday, 11:30 to 12:30 until October 10th, which is until my cycles are active. In my office, every Tuesday, 12:30 to 2:30 from October 11th to December 5th. And I will have extended office hours during exam time and possibly during tests, and that'll all be posted on OWL and sent as an announcement, so don't worry. If you cannot make these hours, do come see me, send me an email, and I will make sure that I book an appointment with you. Post your content questions on the forum. October term test is October 6th from 11 to 1. It's going to address outcomes from cycles 1, 2, 3, and the Williamson article. Okay, multiple choice questions only. Roughly 30, I'm guessing less than 30, but roughly 30. You have an individual and a collaborate. Individual and collaborate. You're going to write an individual exam with, for example, 25 multiple choice questions. Then you will have 10 minutes to get into your groups of 4, which will be your lab group. You are writing the collaborate portion with your lab group. So when you go on to OWL to see which room you're writing the test in, we have multiple rooms because we have so many students, you are going to be in the same room as your lab. It'll be based on your section. It'll say section 036 is writing in health science building. All these sections write in health sci. All these sections write in that side. So you're going to look up your room number based on your lab section number. Why? Because your group will be there, and you have to write the collab with your group. How does the collaboration work? You're going to sit down, you'll be given the same questions. The same questions, there might be a couple of different ones, but the same questions you already did in your individual. And you'll be given a scratch card. So you see question number 1, A, B, C, or D. And you decide as a group, go, I remember this question, I just did it. And then you come to a consensus. Someone says, if everyone agrees the answer is B, then you scratch B. If you get it in the first try, you get 3 out of 3. If you get it in the second try, you get 2 out of 3. If you get it in the third try, you get 1 out of 3. If you get it in the fourth try, which is, i.e., the last one that's left, you get 0. That's how the scratch cards work. Advantage of this is that, number 1, you get to discuss with your peers. what the right answer is, and it provides you feedback immediately. Maybe someone in your group knows the answer and can explain it to you. No, the answer is B because of this. And then when you scratch it, you know what the right answer is because the star is in the right answer. So before you leave the exam, most students have a good idea how they did. Details about the exam will appear on OWL. Today's lecture is that we're dwelling into the DNA, into genetics. My whole life has been genetics. I was always interested in DNA. I never looked at genetics as a cool field to go into. I looked at the DNA as a way to study life. Because every living thing in the world, minus a few viruses, have DNA. And I felt if I can find out more about the DNA, then I can find out more about the life form. Because the DNA codes for everything. It's not the master regulator, which we'll talk about in next term, but it codes. It's the code. It's the program. Computers have programs. That's the program. So if it's programmed to do certain things a certain way, it's all in the DNA. And I always found that so fascinating. How you could, and I manipulated DNA in my master's, I manipulated DNA in my PhD, and I saw the effects, and it was just mesmerizing to me. That's why I went into it. So in this class, we're going to cover mitosis and meiosis, which most of it has been covered in high school and a lot of good explanation in the textbook. So we're not going to go into the steps of mitosis, the steps of meiosis. That's something you should read in the textbook. That was part of the reading. You should know that. How do you get an egg and a sperm? Through meiosis. Prior to that, where do they come from? They come from germ cells. So germ cells, which undergo mitosis, 2N to 2N, 2N to 2N, they then undergo meiosis, become N, and then from that, you get coupling of both of those, sperm and an egg come together to produce a zygote, back to 2N again. That one cell zygote develops into an entire organism. Through differentiation, it differentiates into various different tissues. We can get variation in a population via this process. Where does the variation come? Where do we get that variation, the genetic variation in the population? Where in this process, in meiosis, does that happen? During crossing over. That's why your siblings don't look identical to you unless you're identical twins. It's during crossover that you get that variation in the population. So the genes, your physical genes, your DNA sequence has much to do with it. In next term, we'll talk about the effects of tags on your DNA. But in this class, we're going to talk about the DNA sequence. Mitosis. Does everyone know the stages of mitosis? That's important to know. If you don't know it, please look it up. The other important aspect that leads to this process of producing an organism is cell cycling. Cells have to cycle. And when they go through the cycle, that's how they replicate. So interphase is a part of the cycle, and that includes G1, S, and G2. G stands for gap. What's the gap? There's a gap between M and S. There's a gap between S and M. That's what the gap means. The G1 is preparation to go to S. The S is preparation to go to G2, and the G2 then goes to M. When it's actually doing its job, which is to function in the liver, to function in the heart, to function in the brain, wherever it has a function, it's sitting in G0. It's sitting in G0. Not all your cells are cycling all the time. Do all your cells cycle all the time? No. So which cells cycle, which cells don't? There are certain cells that once they differentiate, they don't cycle anymore. So during embryogenesis, all your cells are cycling. They're in the active cell cycle, and then once they reach to the point of complete differentiation, they don't cycle anymore, or they do depending on what cell type they are. So if they're muscle and neurons, for example, they sit in G0, and they don't enter the cell cycle. Your cells of the liver, they enter the cell cycle because your liver can regenerate. Adult stem cells, which we'll learn about in second term, they regenerate as well. So tissues that require growth, repair, regeneration, those are all the ones that are going to go from G0 to G1 and enter the cell cycle. When they're actually doing their job, they're not in the active part of the cell cycle because they're going to divide. They're going to undergo mitosis. Bacteria divide via fission. It's very simple. If you don't know the difference between prokaryotes and eukaryotes, please look that up. It's important. The main difference is that prokaryotic cells don't have organelles, they don't have a nuclear membrane, and their DNA sits there somewhere. It doesn't have to be always in the center. It can sit anywhere in the cell. So bacteria and archaea divide by fission, the DNA replicates, it starts the origin replication, and then the cell simply divides by fission. It's a very simple process. Eukaryotic cells, on the other hand, divide by mitosis. This is an image of a 3-day-old embryo, and the cell is dividing. We're learning mitosis, cell division, right? Cell division. Okay, so if your skin cells, the lining of your stomach, the intestinal lining, bone marrow, all these things, if they have to grow, they have to regenerate, why do they have to divide so many times? In other words, why do we have 35 trillion cells? 35 trillion human cells. We have more bacterial cells than we do human cells. Why do we have 35 trillion of them? We only have 200 different types of cells. Tissue differentiation, we only have 200 different types. 200 different types of cells translating to 35 trillion cells? Why not just have 5 of each type? 10 million, that's still less than 35 trillion. Why do we have so many? In other words, why do cells undergo mitosis so much? You're learning mitosis. Why the heck is it doing mitosis that many times? Why does it have to divide that many times? Yes? Good, she said the cells will die. But what if I said the cell can grow as large as a golf ball before it dies? Very good. A larger cell is harder to maintain. They need to maintain a high surface area to volume ratio. Okay, we'll talk more about that next term, but have an understanding, there is a reason why mitosis occurs at the rate that it does. There's a reason why we have so many cells. You're learning mitosis, you need to understand why the heck is a cell dividing so much. All right, so let's go on to iClicker and start this question. Okay, I'll just give you maybe 15, 20 more seconds. Commit to an answer, please. Commit to an answer. If you don't know it, it's okay. You need to commit. You need to commit, guys, please, because on the exam, you can't not commit. If you see the answer and then go, ah, yes, that's called hindsight bias. You don't want to create hindsight bias. That's not good training for the exam. Okay, all right, let's stop this. C. So most of the class felt it was C. 65% of the class said C, and 22% said E. The answer is E. The answer is E. I know what every one of you is thinking. Oh my God, this is a trick question. She's going to trick us on the exam. If I wanted to trick you, I'd just put this in the exam and trick you. I'm warning you to look at the chromosomes carefully and how they're lined up. Chromosomes never line up like this during mitosis. During mitosis, during anaphase, the actual centromere splits. So in other words, one sister chromatid on this side, one sister chromatid on the other side. This is anaphase 1. This is anaphase, but it's anaphase 1 during meiosis. Because look at those sister chromatids. They're still held together. They're still held together. Please understand that you need to think slowly on the exam. You have plenty of time. Don't rush it and go, anaphase, next question, and then you go, what? So please be aware of that. Cycling cells. The purpose of actively cycling cells, again, for growth, repair, regeneration. Where can we find actively cycling cells in birds? When Beth talks to you about birds, where would you find them? Stomach lining, gut lining, hair follicles, nails, the bone marrow, skin. Where actively cycling cells in a tree? What are those cells called? Does anyone know? Actively cycling cells in a tree? Meristem. Meristem. Tips of roots, tips of shoots, the cambium layer just underneath the bark. Those are all undifferentiated cells that will not differentiate, so they need to divide, they need to enter the cell cycle. So the cells go from G0 and they enter the cell cycle. Most of your cells are sitting in G0. They're not cycling. If they were constantly cycling, you'd be like the size of the universe. So they're not constantly cycling. When and where might cells be programmed to die? How many of you guys have heard of apoptosis? This is called programmed cell death. Programmed cell death. When would they be programmed to die? Yes?
If it be used to serve the organism like the xylem in the plant.
Very good. Okay, we'll get to that. We'll get to that in a second because I want to say a little bit more about that. Yes? Good. That was, okay, so you guys are going in the perfect direction, which I want to get to. Another reason. So they're saying that they're programmed to do this because that's what they're supposed to do. They're supposed to do this for this process. Good. Okay. Telomere shorten. That's a little bit different. They reach what's called cell senescence. They reach their Hayflick limit, which we'll talk about later. But they don't undergo apoptosis for that. But that's a great thinking. Yes. Okay. They become too large to maintain. In that case, most cells divide. And so they're programmed to do that. They're just malfunctioning, guys. There's a mutation. There's a problem. It's gone rogue. There's a problem. So cancer is a great example where that happens. The cells will apoptose. They will try, but then, of course, cancer takes over and then bypasses the apoptosis pathway and then triggers cell growth, cell proliferation, and anti-apoptosis. So that's how this cancer takes over. But yeah, if there's a damage to the cell will undergo apoptosis, so they don't carry it around. The two answers that these two students gave, often cells their function is to die at the end. So, for example, xylem tissue in plants, to transport water, so it needs to actually go through to that channel. So the cells are programmed, once they're done their function, they die, so that it can become a xylem. Your fingers, which is the student example I gave you, during embryogenesis, your web-like fingers that become appendages, they actually die. The middle parts die, undergo apoptosis, so you can have these appendages. So there are normal functions that lead to apoptosis because that's required for the function of that particular process. Checkpoints. Where would you find checkpoints? At G1S, G2M, and metaphase in mitosis. That's where you would find checkpoints. Why do you think there's a checkpoint at those places? If you had to put checkpoints, where would you put them? Would you agree that that's pretty much where I would? But you can't put checkpoints everywhere. There, for the most part, believe it or not, there are some checkpoints everywhere. But these are the big players. And the biggest player in this, the biggest one, if you search the literature, is G1S. G1S is the biggest one. So why G1S? You thinking? Yes. Good. These are critical points. Why are they critical points? Why is the G1S a critical point? Very good. Would you want to replicate your entire genome before if there's a problem, are you going to say, you know what, how about we invest a lot of energy and replicate the entire genome, and then we can go undergo apoptosis? No, how about we check it before? And what if they can't undergo apoptosis? Now you have duplicate, then you have two daughter cells with problems. So yes, that's a big checkpoint. G2M, why undergo mitosis? Let's not undergo. Are we ready for mitosis? Are we ready to actually physically divide the cell into two? Because we don't want to create two problems. We already have one. And metaphase. Why metaphase? Why not prophase? Yes. Exactly. They need to be lined up correctly so that when the sister chromatids separate and all those spindles, they have to be attached properly so that when they separate, they actually pull them apart so that they get even distribution of chromosomes. If you get uneven distribution, for example, during meiosis, during the production of a sperm and an egg, then you can get Down syndrome. That's how you get Down syndrome. So you get an extra chromosome 21. You don't want that to happen, so that's another checkpoint. Where would you find DNA in a plant cell? You would find DNA in the nucleus, in the mitochondria, and the chloroplast. The DNA in the nucleus is different. The DNA in the mitochondria and the chloroplast is different. Mitochondria and chloroplasts are descendants of prokaryotic cells through the process of endosymbiosis, and so they have prokaryotic DNA. It's circular. Not all of them have circular ones, but for the most part, it's circular, and it's very similar to that of the bacteria. And that's one of the key evidence showing that this happened by the process of endosymbiosis, is because of the type of DNA that these two organelles have. And the nucleus has eukaryotic DNA. This is important. So I would strongly pay attention. This concept seems pretty simple to understand, but as soon as students look at an exam question, this is where the vast majority of confusion and the vast majority of incorrect answers come from. A genome is all of the DNA sequence in one copy of an organism's chromosomes. N represents one copy of all of an organism's nuclear chromosomes. Just one copy, not every single chromosome in there, just one copy of it. So we have 2 copies. So our N is 23, our total number of chromosomes is 46. Our N is 23. We have 23 different chromosomes, but we have 2 of them, right? So N is one set, we have 2 sets. We have 2 sets. So our ploidy is 2N. C represents the amount of DNA in one set of an organism's nuclear chromosomes. And C is the amount of DNA. So this is measured in picograms or base pairs. So C is how much DNA do we have in picograms, for example, in one set. So how much DNA do we have in those 23 chromosomes? How much does it weigh? That is C. So please understand the difference between N and C and ask yourself, how does N and C change throughout the cell cycle and during mitosis and during meiosis? How many of you are very comfortable between the difference between chromosomes and chromatids and what defines a chromosome? You all should know that that's what the DNA looks like in G1, and then when it replicates via semi-conservative replication, in S phase, you get that formation right there, okay? And that, during metaphase, the DNA gets condensed. It gets condensed, and that's why when you look at these karyotypes, and you look at the DNA in the microscope, they look at it in the metaphase phase, because that's when you can actually see it in the microscope. You can actually see the structure of the chromosome. It looks like that. So that top part right there, that's a chromatid. That is a double-stranded helix. That's not a single strand of DNA. That's still a double strand. DNA is always double-stranded. But that top part is a chromatid, and the bottom is a chromatid. They're sister chromatids. They're sister chromatids. This whole thing, that whole dark black thing is called one chromosome. A chromosome is defined by the number of centromeres, not the number of strands, the number of centromeres. So, when you have a chromosome 1, it has a centromere. Then it replicates during S phase, but it shares the same centromere. So chromosome 1 is 1 chromosome, and then when it replicates, it's still 1 chromosome. But it's 2 sister chromatids. Have to keep that straight. 1, 1, 1. They're all 1 in each case. Starts off with 1 in the cell. DNA synthesis happens. You get 2 sister chromatids, but you still have 1 chromosome. I didn't come up with this. I don't think this is the best way to do it, but this is what it is. So it's based on centromeres. There's still 1 centromere. Then, when it splits after anaphase, it's still called 1 chromosome. I know, this is going to confuse you, but don't let it confuse you. It's laid out very clearly. It's always 1, 1, 1. Count the centromeres. So when you look at, this is important, a karyotype, and we ask you, how many chromosomes, how many chromatids, what's the N value, what's the C value, you got to be able to do it. So here's a human karyotype, and again, vast number of confusion happens with this. Just completely lost. Don't be lost. If you have questions, come see me, and I hope that this lecture is explaining it in a way that's very clear. So look at this carefully. This is the karyotype of humans, 22 autosomal 1 sex pair of chromosomes. That chromosome 1, this whole picture is during metaphase. You can't capture a picture like this. So someone took pictures where all the chromosomes are all mishmashed. They took it all and they arranged it like this, or a computer did it, and they took a photo. Someone did this. This is not what it looks like. Someone did this. But those chromosomes, that's how they look like under the microscope. They just arranged it for you so it looks like that. That's called a karyotype. This is a human karyotype. If you look at the chromosome 1, you have one from your dad, one from your mom. But look at that one strand. That one strand, that's a pair of sister chromatids. You can't see it. The resolution is not there. You can't see it. It has to be because DNA replicated. It hasn't broken apart yet. So it's still in metaphase. It's gone through S phase. So each one of those is 2 strands, but it's held together by 1 centromere. Got that? This is really important. This is pivotal. It's going to be on the exam. It's going to cause confusion, but I know you guys can ace it. All right? I know you can do it. The rest of the slides is in the textbook. This is redundant information for me just to sit here and just tell you, this is a microtubule, this is an intermediate filament. Please read up on it. You're responsible for this. Don't go crazy with it, but please understand all the terminology that's discussed in the rest of the slides, all right? We'll see you next week.